Hello, everyone. The paper we chose is the potential anti-cancer effect of helolin as a natural bioactive compound on the growth and telomerase gene expression in breast cancer cell line. Breast cancer is the highest prevailing form of cancer in women. It is second to lung cancer as the leading cancer that causes death. As most people know, the various forms of treatment for cancer are often harmful to healthy human cells. An alternative form of treatment that is not toxic to healthy human cells would be immensely valuable. The researchers in this paper believe that helolin can serve as that alternative form of non-toxic treatment. Helolin, which is depicted on the right, is a small organic molecule produced by Arnica chimizones and Arnica montana flowers that is not necessary for the growth, development, and reproduction of the plant. Through a long history of consumption and various research studies, it is known that helolin has anti-cancer and anti-inflammatory therapeutic effects. Telomerase is a protein that allows cells to continue reproducing and growing beyond their normal life cycle. It is typically inactive in healthy human cells, but it is active in 90% of breast cancer cells. Human telomerase reverse transcriptase, known as hTERT, is one component of the telomerase protein that is essential for its functionality. There are many studies that focus on inhibiting telomerase as a way of treating cancer. This study focuses on using helolin as a non-toxic form of treatment to treat breast cancer specifically. The researchers of this study sought to answer the question, how effective is helolin extract at inhibiting telomerase gene expression? They hypothesized if helolin inhibits hTERT gene expression, then helolin extract would have a major use for anti-cancer therapy in breast cancer.
Some of the materials that they used were cell medium, which is used to grow the cells, and was treated with fetal bovine serum and some antibiotics to prevent growth of other bacteria or cells that may be introduced. They also used an RNA extraction kit to be able to take out the RNA from the cells that they grow from their cultures. And then after that, they used a green real-time PCR mix from a company in mixed with MTT powder, which is used to test for toxicity of helolin, I think. So first part of the methods was the cell culture and the cell cytotoxicity. So the cell culture, as I said before, was prepared with some antibiotics to prevent outside organisms, and had a pH balancer added to it. So it makes it easier to grow the cells. The cells were also grown in a CO2 incubator, which you can think of it as like a bread proofer or an egg incubator, where they controlled the environment. So they set it at about 5% CO2 and 55% humidity. This was done across 96 plates to make around 2,000 cells each cycle. And these cells were treated with 5 different helolin concentrations from 1.0 to 5.0 micrometers. Then this was tested across 3 periods, each of them lasting from 24 to 72 hours. And there's also a negative control that did not receive any treatment, and a positive control that had a drug that they knew would have the proven effect that they were looking for. For the RNA extraction, the cells were centrifuged to separate the bulk of them from the medium so they could then extract the RNA from the cells using the purchased kit. And then once they were able to purify the RNA, they used primers from their cDNA kit to synthesize cDNA. cDNA is just complementary DNA. It's the same as DNA that we have everywhere else. It is synthesized or made from RNA. RT-PCR, which in the case of this experiment is real time. They used it to isolate the H-TERT gene and the beta-actin gene from the cDNA that they had synthesized. And this was done across all controls and the treated cells. The primers that they used were synthesized or made by GenBank. And the statistical analysis that they put their data into was in GraphPad Prism 8.4.3. And then the difference of like the H-TERT expression levels were like analyzed through there. And if they had a difference of about 0.05, it was a substantial difference. So that would be noted.
Results from this test showed that halonin reduced the cell growth, and this was based on a time and dose-dependent manner, as indicated by the results noted. Additionally, the real-time PCR test demonstrated a significant decrease in the H-TERT gene expression in the cells that were exposed to halonin, and this was also, the results of this were dependent on the time and the dose or dilution concentration pattern across the different treatments in comparison to the control group. In general, and overall, the therapeutic and cytotoxic effects of halonin were dose and time dependent. So they vary based on the dilutions of the different treatments and how long the treatments were studied across. And these are just 3 graphs with varying data points for the different time periods ranging from 24, 48 hours, and 17 hours treatments, with the y-axis displaying the cell viability, and then x-axis is the halonin concentration in microliters. In reference to the H-TERT gene expressions, the results indicated that halonin actually hinders the growth of the cells in a time and dose-dependent manner, and the data also supported this as well, with values of halonin at 24, 48, and 17 hours. And reference to the micrometer data for this, it was 4.69, 3.67, and 2.23 respectively. And so the data from this experiment supports that the halonin extraction had inhibitory high effectiveness on mRNA gene expression of H-TERT, which is a potential anti-cancer remedy for breast cancer, which is a significant thing to note for the experiment purposes. And so the growth of cancer cells after the treatment, it was found that visibly their cells were morphing in appearance, and that can be seen in the 2 visuals on the bottom half of the slide. And particularly, there was a note on focusing the growth rate in between the control and the treated halonin groups. And so with the microscope, it was noted that there were alternations in the cell morphology and appearance, and there was also a decrease in cell concentrations and numbers in the cells that were exposed to halonin, which can be seen in the picture on the right in comparison to the control group that was not treated.
What we learned from the results were that halonin hinders the growth of the T47D cells, and the visible cell changes were noted as from the previous slide. But what does this mean? This means that using methods like the RT-PCR, it can help us reduce the number of the cancer cells because the halonin hinders the growth of the cancer cells. And, by implementing the halonin into the cells, they can be drastically reduced and degraded. And we do agree with the results of the experiment, because the statistical data showed that the growth of the cells decreased with the increase of the halonin inside the cells. And then in conclusion, the results of this experiment showed that helanin extraction had an inhibitory effect on the hTERT gene in breast cancer cells. And the experiment also showed us that the effects of the helanin were not only dose dependent, but time dependent. And the statistical data also supports this, as the graphs showed on the previous slides, show us that the higher the dose, the less expression. And the data showed the helanin extraction had a high inhibitory effect on the mRNA expression on hTERT and could one day be used as a solution to breast cancer. So the study shows us that one day in the future, helanin could be used inside someone to reduce the expression of the breast cancer cells and essentially be a remedy for breast cancer, which is very, very innovative and very revolutionary because it's not been done before.
The researchers suggested that the next step for this research is a preclinical in vivo model to confirm the effectiveness of helanin in living organisms such as mice. Once the effectiveness is confirmed in an in vivo model preclinical study, the next step is a clinical trial to test the effectiveness in human patients with breast cancer. Another possibility for the future of this research is to test the effectiveness of helanin on other forms of cancer, starting with a preclinical in vitro model study similar to the study we've explained today. Here's our references, and that's all we have for today.